Hello dear friends, all of you good morning. Today we are going to discuss about the RNA interference. Before understanding the RNA interference, we have to know about the DNA and we have to know about the RNA. The DNA is a double stranded and RNA is a single stranded, and this RNA will forms the protein. Then question is how it forms the protein. The DNA, which is a double stranded, it having the two polarities 3,5 and 3,5 and opposite to each other. When it forms the mRNA, that process is called the transcription. Whereas when a RNA forms the protein, that is called the translation. This transcription occurs in the nucleus, as we know, and this translation occurs in the cytoplasm. Now, try to understand these two strands. If I will make it a straight line, suppose it is a 3,5 polarity and it is a 5,3 polarity. Suppose it will forms the mRNA. What strand it will forms? 3,5, which we call as a template, and this is called the coding strand. This coding strand does not code any strand, but template will be strand, double of the strand. And when this template will code what, it will forms the complementary of the RNA. Then the complementary will be, because it will be this template strand will forms the complementary, then the polarity of the mRNA is 5,3, and the codons are present, and these codons are forms a protein. But now you try to understand, only one strand of DNA forms the protein. If suppose you imagine two strands of the DNA, when it will forms the RNA, what will happen? Now see, it is a one strand and it is a one strand. It is a 3,5 and it is a 5,3. And when the two strands will form, then the double stranded RNA will form, dsRNA. Then what will be the result? If the dsRNA means double stranded RNA will form, it won't form any protein. Why? It will not form any protein, no protein formation. Why? Because if the single RNA strand, it contains the codons, and that codons will reflect, that will helps in the protein synthesis. When these two strands of RNA, when they will forms protein, it won't be happen, because all are become the complementary and the codon will unable to form the proteins. That's why there is no formation of the proteins. Then one question is, if the protein will not form from the DNA, what is the use of the DNA? Because see, imagine if this DNA is a harmful parasite, then we have to stop the protein synthesis, because in the parasites this protein formation is causes the toxic to us and forms the symptoms. If we will able to stop the any parasite, we have to form the double stranded RNA. Are you clear? Now, what we understood until now, we understand that if the double stranded RNA will form, there is no chance of protein synthesis. How it will form? When the two strands of RNA will unable to and cease the protein synthesis, because the codon are expressed to form the protein or amino acids, as we know. Then one nimatode is there, name is Meliodogyne incognitia. What is this Meliodogyne incognitia? It is a nematode. And what is the problem with it? It will infect to the tobacco plant. When it will infect to the tobacco plant, what it will do? It produce the protein. How this organism will produce the protein? Because it having the DNA of whose DNA of Meliodogyne incognitia, and it will forms the mRNA and it will translate to the protein. This protein is toxic to the plant and it will reduce the production of the tobacco plant. Then how we will stop this mRNA? We want to stop the protein synthesis, because it is a toxic for the plant, for the tobacco plant. Then how we will stop this mRNA? We have to stop this mRNA by forming the double-stranded RNA. Then how it is possible? Now we will take a bacteria that is called azotobacteria. For your clear information, the azotobacteria you have to know because it is genetic engineered. Every bacteria having one plasmid, which is an extra nucleus in the bacteria, and this bacteria which is present in the agrobacterium is called the tumor inducing plasmid, because plasmid is every bacteria having the plasmid, and this plasmid is Ti plasmid. Why it is called Ti plasmid? Because it produces the tumor in the eukaryotes. It is only the bacteria which is forms the tumor in the eukaryotes. All the bacteria are prokaryotes, but this bacteria having the ability to produce the tumor in the eukaryotes. Now you will take a small gene, it having a small piece of DNA is called the tDNA. What is this tDNA? Transfer DNA. Why it is called transfer DNA? Because when it will infect to a eukaryotes, the whole plasmid will not go inside the eukaryotic cell. Then how it will infect? It will send a small piece of DNA which is called the tDNA, transfer DNA, and infect the eukaryotics. But now what you have to do, try to understand everyone very carefully. This DNA we have to isolate from the Mylidogyne incognitia. Let's say it is the DNA of the Mylidogyne incognitia, and it will transport to the agrobacterium. When it able to transport to the agrobacterium, now the new plasmid which is formed, it doesn't have any parasitic effect. In place of that, we have to manipulate like that. This plasmid forms the double-stranded RNA. Where it will form the double-stranded RNA? Inside the tobacco plant. This tobacco plant, inside that, this RNA will form in the tobacco plant. Then what is the benefit for us? When this Mylidogyne incognitia will attack to the tobacco plant, then what it will form? Already in the cytoplasm there is a dsRNA, double-stranded RNA. Now the Mylidogyne incognitia infect to a tobacco plant, and when it will infect to a tobacco plant, this RNA become entered, it will send the DNA, then DNA will form the RNA. When this RNA will enter, which is already the dsRNA is present, immediately it forms a complementary of this RNA, by which it will stop the protein synthesis. Protein synthesis will not occur. When this protein synthesis will not occur, then what will happen? That will, the Mylidogyne incognitia does not able to cause the infection to the tobacco plant. Make it very clear once again. That Mylidogyne incognitia is a nematode which is infect to the tobacco plant. Infection means, what does it mean? It means that the DNA forms the mRNA which is form a protein which is toxic. But if we are able to form the double-stranded RNA, it will stop the protein synthesis. That means we are the microorganism or any nematode unable to infect the plant. For to form the double-stranded mRNA, we take the help of the azotobacter, isolate the plasmid, and that isolate the plasmid, it having the tDNA. We have to manipulate it like that. When it will infect to the tobacco plant, it will form the double-stranded RNA. And this double-stranded RNA present in the cytoplasm of the tobacco plant. When the Mylidogyne incognitia affect to the tobacco plant, what it will do? Immediately it will form the complementary of the RNA, by which there is no protein synthesis in the Mylidogyne incognitia, which is formed by the Mylidogyne incognitia, by which the infection of the tobacco plant will get stopped. It happens by the formation of double-stranded RNA, which is not allowed to the protein synthesis, the protein which is a toxic for the plant. That is a mechanism. RNA interference is a mechanism by which we can prevent. It is a defense mechanism in the eukaryotics. Thank you for watching the video.